It is my pleasure to introduce Greg Thompson, Professor of Engineering at the University of Alabama. Professor Thompson has published over 200 peer-reviewed papers in his fields of expertise, which include phase transformations, deformation mechanisms, and thin film growth. He joined the University of Alabama's Department of Metallurgy and Materials Engineering in 2003. In 2018, the University of Alabama system recognized him as a distinguished university research professor. He currently serves as the Alabama Analytical Research Center Director, overseeing a multitude of electron and ion microscopes for the university. The title of his talk, The Use of Precession Electron Diffraction for In Situ Cross-correlative Microscopy. And now, here's Professor Thompson.
Hi, everybody. I first just wanted to show my face before I put the webcam out and say thank you so much for your time to join this webinar. I'd like to pass on my thanks to Nanomegas and to Robert for the invitation to contribute to this series that they're performing today, talking about PED and its application in material science. My talk, as Robert said, is a third in a series that they've done. And what I hope to convey over the next 30 to 40 minutes is how we've used PED, precession electron diffraction, in a variety of different material systems to elucidate and understand the underlying structure that governs properties. So it's really meant to be kind of a high or more overview view of how the technique has been applied, particularly in the research that I do in my group. And with that, I'm excited to share with you what we've been able to do. Some of the stuff has been published. Some of the data I'm going to share with you is actually hot off the press, if you will. It's in the peer review process. And again, I want to thank you for your time, and I hope this is informative to you and how PED has been very impactful in the type of research that we've been able to do at the University of Alabama, and for the individuals and students and postdocs that have had the great opportunity to work with. So this time I will remove myself and start the talk. So as I indicated, I'm Greg Thompson, and a lot of the work I'm going to share with you today is from some former students of mine, Justin Bronze, who's now moved on to Apple, Cheng Go, who's a postdoc at Oak Ridge National Laboratory, and Rhett Zhou, who is now a Humboldt Fellow, which I'm very proud of for him, at the Max Planck Institute for Iron Research in Dusseldorf, Germany. And part of this work, particularly with Cheng Go's work that I'll share with you, I worked with my colleague Lynn Lee and her student, Yuchong Gu, on modeling aspects. And then I'm going to motivate the work that we did, what Justin did, with some colleagues I had at, or still have, I should say, at Sandia National Labs, Khalid Hattar and Brad Boyce. And all this work has been supported by various funding agencies, whether it be Department of Defense, the National Science Foundation, or the Army Research Office. And I would like to acknowledge them and to thank them for giving the students and myself the opportunity to work on this project. Let's see, next slide. Let's see. There we go. All right, so what I want to start out with is I'm going to talk more on the application sides of PED, but I recognize that some of you may have not attended the first two webinars in this series, and so I just want to quickly recap how precession electron diffraction works, so that its application, what I'll show in the remaining slides, will make hopefully more sense. So for those that know this, I apologize. Hopefully this will be a nice summary though. In short, we take an electron beam while we're in the transmission electron microscope, or TM, and we process it. And for our group, we tend to process this between 0.1 to 0.3 degrees. And as we rotate the beam, and this is a full precession, as you can see, with the beam processed above the sample, and then we fully process it at the bottom, and we see the diffraction pattern. The advantage of processing the beam is that it actually helps average out the dynamical effects of electron diffraction. And so in that sense, then it allows the diffraction intensities to be much more uniform. So obviously, if you've been involved in TM, you know that the intensities of the diffraction pattern can change as the foil thickness. And so this way, by processing it, makes it more kinematical in nature. So that allows us to obtain the patterns we need. The other advantage is by processing it, the electron beam, it's able to cut through more of reciprocal space. So we're able to get more diffraction spots, which then allows us not only have more uniform intensity, but more diffraction reflections to increase our confidence in how we're going to index that diffraction pattern. So processing beam has been, is really key to how this whole process works. Now, if we take that beam and we raster it, in a series of incremental steps as we go across our foil, then we're able to collect a pattern at each of those points. And so typically for us in our group, we typically make step sizes between 3 to 5 nanometers. Of course, that's the finest that we can do. Or you can, obviously go up higher. But what you do is you collect then a diffraction pattern at each of these spots along the foil, and you've captured those images. You then take the diffraction pattern that was collected, you compare it to a template that allows you to identify the orientation and also the phase of the structure. And with that, it generates what is called, or the terminology is still developing a lot in the field, but PED is kind of the method of collecting the data. But then once we collect it and we identify those patterns, some of the more names that are becoming more familiar with, for example, is automated crystallographic orientation mapping, or ACOM. And that allows us to determine the phase. So if it's FCC or BCC or some other crystal structure, as well as the orientation of each of those phases. And what's really, and which we take quite a bit of advantage of, is not just the texture that we see, but also the relative misorientation as we go from one grain to the next. We see that diffraction pattern change its orientation, and in doing so, we're able to determine its misorientation or the grain boundary type. And we use that quite a bit in the work that I'm going to share with you. So this is generally in a very short recap of how PED works and how we apply it. So I want to kind of begin the talk by where we started with PED. Not that I need to follow a chronological order, but I think it kind of indicates how powerful PED can be in understanding outstanding questions of material science. And so this first one isn't necessarily in situ, but I think it sets the stage of what it can provide and how applying it then to in situ, for example, deformation or heating experiments, can provide a lot of really good insights. So in 2005, there was a report that in nanostructured or nanocrystalline copper grains that were indented at liquid nitrogen temperatures, that some of the grains grew. And that seemed very peculiar because a lot of times when we think of grain growth, we think of it at a high or elevated temperature. And so they induced this grain growth through a mechanical indentation effect. And then they went in the TM and then they just did bright field imaging and dark field imaging and really say, well, some of these grains have grown. And so this is kind of captured here in these pictures, as well as the quantification they acquired of the grain sizes from the TM images. But it really left the question is, how does this occur? Because at cryogenic temperatures, we would assume the mobility for grain growth would be quite low. That said, working with my colleagues at Sandia National Lab, we then began to understand, do our own set of experiments. And so what we did is we made 3 types of copper films, one that was nanostructured and nanocrystalline, and then 2 others that both had a high density of twins, but the film was oriented in either a 111 direction or a 100 direction. So we took those films, we submerged them in liquid nitrogen, we used a custom-made indenter that you can see shown here, and then we indented the sample. And then after indentation, we removed the sample and then performed a fib lift out of a TM foil from the indent region. And so this is captured here in these papers for those that are more interested in some of those details. Now, of course, there's some questions that occur that the sample heated up and you indented it, and did the growth occur at temperatures? And those are actually problems I'm solving today with another collaborator of mine, Eric Homer, on trying to do these experiments in situ conditions. But with that, we then were able to fib out with the foil, and in the nanostructured film, the nanocrystalline film with no twins, we really didn't see any type of grain growth. This was the parent material. We see that the grains are actually quite smaller, where in the pile up or flank region, and then under the indent they were quite small. But what was kind of nice, we can identify in some cases the grain boundary types through PED. And so this is kind of where maybe knowledge was located, but through having the ability to use PED, we're able now to look more closely at the grain boundaries. And so in this case, when we looked at the nano-twinned 111 sample, we can see that, again, under the indent, it was compressed. In the pile-up region, we see the grains were quite large, and so we do see the cryogenic grain growth. And what was interesting is we're able to now identify the grain boundary types. And in this case, in this pile-up region, we did not notice any type of special character grain boundaries. They were all high angle grain boundaries, and there was no twins. And similarly, when we indented our nano-twinned copper film that was oriented in 1O direction, we again see this grain growth in the pile-up region, and we're able through PD to be able to go in and identify the boundary types. And again, we notice that in the regions where there's a lot of growth, we don't see necessarily a lot of twins. There's some present, but in this case, we didn't see any. So what this really gave us insight is that the twin structure in this copper sample must be very important. And we're able to use PD to study it and quantify that because we're able to actually measure that in the nanoscale regime, where typically other techniques prior to PD, such as electron backscatter diffraction, may be limited to 100 nanometers, which would be far more coarse than actually the size of the features we want to see. And so by having the PED data, we're able to then go in and quantify the grain size distribution through these cumulative area fractions, both in all three regions, the parent region, which are all about the same, the pile up, the flank region where they grow, and then the indent region right under it. And what we can see is the indent region is that the stresses are so significant that the grains are really, really refined. But we're really able to show very conclusively for example, in the 100 oriented nanotwin structure, we have significant grain growth into this region. And so now recognizing through having the ability to identify the type of grain boundaries that twins must be an important contributor to the behavior, and the fact that we don't see the twins in the grain growth regions, it gave us a lot of direction to recognize that the potential mechanism for this cryogenic induced grain growth may actually be a detwinning mechanism that has been proposed by, for example, Chen and others of what may be occurring. So basically, under the mechanical load, that the twin may be unzipping itself, and then in that process of unzipping itself, the grains now appear larger. And so this, again, what I wanted to get across with this to motivate the rest of this talk was just recognizing how PED is the ability to ascertain and quantify these small scale grain structures really gives us the insights of how mechanisms occur. And so with that success, we were even able to, knowing the differences in twin densities we see, where we see a few twins and no twins, we're able to even go further to understand that behavior, because we recognize that in some loading conditions, there's the Schmidt factor would be able to produce the type of force onto the grain boundary twin to cause a DD twin. So we're able to even further understand what's going on between the two types of samples. So with that, I want to now move into really what this talk is really about for the rest of the session, which is the use of PD in cross-correlative microscopy techniques, and when possible, its use in situ TEM experiments. And the first one I want to share with you is some work that we did again on copper, where we indented the sample with a PI 95, well, I guess it's Brooker now, but it was the time Heisitron indenter. And one of the nice advantages of this type of indenter is that we can load it, but it also gives us a load and displacement data, so we're able to get quantitative information that we can collect in the TM. And so this is kind of just an image and a schematic of how it works. We have a sample which has been sputter deposited onto a silicon wedge, and then the transducer comes in and indents the sample. Now, one of the things that we do to help improve the electron transparency of our samples, we deposit onto this wedge by sputter deposition, and then by going into a fib, we're actually able to then thin the foil to make it as uniform as possible in thickness, and as well as to increase the area of electron transparency. So we deposit on the film, we go into the fib, we can quickly make the sample even more electron transparent, better quality, and then we are ready to go and just directly indent it. So in this case, the sample preparation is not that arduous. Okay, so with that, let me just show you, this is the experiment I'm going to discuss with you. Here's our copper foil. We can see that we're loading the sample, and in this case, we see, a lot of bend contours that are developing. We see dislocations that are now coming into contrast. We can see contrast is forming under the indent. We now see dislocations appearing and emanating out of the grain boundary that's separated between these two columnar grains. And we're obviously just collecting an enormous amount of contrast detail of how the material is deforming in the experiment. I'll let this run its course. And then we unload it. We can see that this bend contour comes back together, but we know there's a boundary there through the experiment. We pull it out, and of course we plastically deform the sample. So we have our indented crater. It's located here. We can even see some dislocations up here on the surface that were probably there before, and were just brought into contrast through the indenting process of the sample. So historically, this would be the type of images that you would collect through an in situ experiment. They'll be bright field, you can do some dark field imaging, but this would be historically what you would see. And what I'm going to show you in the forthcoming slide is how PD really helps unravel and unveil a lot of other features that are lost or not recognizable in the bright field images. So one of the problems of, for example, this holder is that you only have an alpha tilt. So in order to bring out other features, you'd love to have alpha and beta tilt in the samples, but you're really limited to one tilt. So that's going to really limit the type of features you're going to see based upon what the contrast you have. And so let's just walk through then what we actually see in a bright field image and then compare that to what we're going to see in a PED scan. So over here we have our load and displacement curve. And then of course our alphabeticals B through G shown here are these various snapshot images at each of those conditions. So in the B image, this is just before we get indenting. We can see a columnar grain structure. We have morphologies that look to be potentially twin structures. We know copper twins quite easily, but we don't really have the diffraction evidence at this point to verify if that's really a twin boundary or twin structure. Then once we get up to point C, we see that the slope is occurring. It's starting to look like it's yielding. We even notice, for example, that the bend contrast that we see on the right side of our indenter is not symmetric, as you can see over in this image, suggesting that these grain boundaries may act as means of resisting deformation, producing the type of increase in load that is needed. But then right after C, we see a load drop. And again, if we go back to our bright field images, we can see this contrast, that band contrast that seems to be kind of to the right, is now much more uniform. Under the indent sample, we can now see that features that we didn't see before. We see these dislocations that appear to maybe be coming out. This boundary is much more evident as before than what we do before. And as we continue to load the sample further and further, we see increased diffraction contrast. We even see at this point, for example, the grain boundary and what appeared to be a single contrast band being split into two, the onset of dislocations, which you saw in the movie that seemed to be emanating out of the boundary. And then once we're finally done, this is what we're left to look at. And so now we're really stuck with trying to decipher what has transpired microstructurally in each of these sequences, particularly in the post-mortem construction. And clearly, a lot of the plastic deformation, there's a lot of deformation that seems to be masking the features and the grain structures that are actually in the sample. And so this was really remarkable to us because we had just gotten the PD platform. And so what we did is we did our PD scan. Of course, we're not able to collect a PD scan in real time because it's a serial process. But what we're able to do is to collect the pre-indentation scan and then the post-indentation scan, and then use this to try to help us understand what transpires in between the two conditions. So in this grain orientation map, we clearly were able to, at this point, identify the grain boundaries. In this case, we see that at the top of the surface, there was some, I guess we'll say these are much more refined grains in here. So the indentation process obviously produced a much more fine grain structure under the indent tip, which was clearly not visible in the bright field image. We can also even identify the texture change within the grain, which is actually quite interesting, because if you look here, that this grain has a yellowish tint to it, and then after indentation, this grain is going from green, which is predominantly a 100 texture, to yellow, which is 102, and then to this orange texture, which is 103. And you say, well, maybe the foil itself bent, and that is what is creating this texture change. But again, having PD, I'm able to look on either side of this grain and notice that, yes, the extent of the color changes is somewhat varied, but it's still green, indicating these grains haven't really bent, for example, as much as this guy. And so this gives us a lot of ability to then have confidence, and that'll come out in some of the work on some amorphous crystalline multilayers I'll show later, of being able to determine if grains are rotating or what's occurring at an individual grain basis. But using the PD scan, we're able now to get an idea of the grain structure quite clearly. We're able to now take the image quality map that you can see, and we can overlay it with a series of grain boundaries from the PD scan. So here we're able now to choose, okay, we have a lot of higher grain boundaries. We have sigma 3 boundaries, as we can see here. This is actually turns out to be a twin boundary, and sorry, sigma 3 boundary, excuse me. And then even in this case, which was really interesting, this was a sigma 3 boundary, which after indentation became a sigma 9 boundary, and that has been previously noted to occur in FCC materials as a phenomenon that's been observed in FCC materials, which has been explained by interactions of grain boundaries and triple junctions. So again, having this PD ability really allows us to get into and understand what's going on. Even in this case, we can see under indentation, this boundary length, the sigma 3 boundaries, actually increased its line length, which is usually increasing boundary length is energetically unfavorable, but it suggests that this boundary must be a low energy boundary and a preferred structure that actually occurs during the deformation process of a copper sample, as you can see here, because it obviously increases its structure. And then finally, we can do a local orientation spread using the PD data. And prior to indentation, a very minimal local orientation spread, but of course, after indentation, we clearly see localized orientation deviations. And in this case, most of it centered under the indent, which would be linked to our approximation of the geometrically necessary dislocations that are needed to accommodate the deformation. So, and I think the next slide, yeah, really kind of illustrates what I really think PD can to help us understand is that here is our bright field image, and then our PD image, and you can really see in the PD image, really clear distinction of where the grains are, that not only boundary types, but where they are that may be lost. And then after you indent the sample, where the diffraction contrast is so strong that you really, if I hadn't done PD, I would not be really aware that under the indent that it caused a formation of small grains to form, maybe that's into the sample. But through PD, we clearly are able to see how these, if you will, coarse grains became refined under the indent. It's very obvious. And the other advantage is that you say, well, maybe these are artifacts because the foil, these are always issues in bright field imaging with these type of experiments, because, well, maybe the foil's bent and we're seeing one pattern on another, but by having the individual diffraction patterns along as we serially scan this area, we're able to really distinctly see very clear, crisp patterns, indicating that these are really grains and are not an artifact. And so I really think this picture brings together what PD can do in your use of cross-correlative microscopy techniques, in this case, in situ deformation. With that, then, we've been able to continue this work of using in situ deformation, but now we've moved into looking at a composite structure. So it's really centered around the idea of metallic amorphous materials. These are materials that have high strength, but also exhibit very high elastic limits. And so they are very attractive for a variety of structural applications. And so this figure kind of illustrates where these bulk metallic glasses reside in a comparison to other materials, for example, steel, silica, or polymers. And so it finds this really nice niche in this high strength, high elastic limit. The problem with bulk metallic glasses is that when they're under deformation, they can exhibit shear localization, shear banding, and that leads to catastrophic failure in these materials. And so when they fail brilliantly, if you will. Now, a mitigation strategy then to stop this catastrophic failure of shear banding is to form crystalline phases within the material. So making a composite of a crystalline bulk metallic glass material. And a lot of times what people will do is they'll take all these variety of elements that give rise to bulk metallic glass and then anneal it such that it will start to crystallize. And you can see these crystal-like dendritic phases shown here. And then these have obviously been able to show significant improvements in the survivability of bulk metallic glass. The issue is that I guess from a scientific point of view, what's difficult is it may be difficult to control the spacing between the amorphous and crystalline phase, for example, between the dendrites and then in between the dendritic arms, to really understand what governs deformation, whether it be the amorphous phase, the crystalline phase, and what is the interrelationship between spacing and volume fraction. So one way around that is to understand more from a fundamental perspective is to grow these type of bulk metallic glasses as crystalline layers and amorphous layers, whereas you change the layer thicknesses, you can really tweak in the variable that you want to study experimentally. And so this is just a paper from Kim and others that have made these multi-layers, did some various tensile testing of materials as a function of the layer spacings. What we've been able to do, or continue to do, is that we've now made a similar multi-layer, as you can see here, and we wanted to understand that deformation in real time and correlate that with PED, particularly understanding the crystalline behaviors response, because PED is based upon electron diffraction. We have primarily just used it for the crystalline phase, but I know that Robert had a seminar, I think a couple weeks ago, that talked about short range ordering and other attributes that PED type techniques may work on. And so there's even avenues of development here that which I won't be able to discuss in my talk, but I'll focus on the crystalline layer. So we have our amorphous layer here, we have our crystalline layer, an amorphous layer, crystalline layer, and of course our amorphous layer, and then our substrate. And so we can watch here as we go through this. We can see a lot of again contrast changes that are going on. We can see correlate this to maybe a pop-in event that we see here of a shear band. And we now have to decipher, well, what is going on in the reactions? Why are certain grains coming into contrast? Why are some going out of contrast? Why are some grains maybe changing their size or their shape? So again, what we do is we'll compare it to a series of still images that what we would normally do in a in this type of bright field experiment with different areas along the load and displacement curve. So again, we use the same idea that the numerical B through G represents different snapshots along here. T1, 2, and 3 represent specific features or grains that I'd like you to pay attention to of what's going on. What we notice is that once we first start to load, and then by the way, the top layer of crystalline is the one closest to the indenter, and then when I say the bottom crystalline layer, that's the one furthest from the indenter. And so what we can see here is as we indent the sample, upon contact, upon really deformation, the only feature that we really see having showed some significant contrast change seems to be this region here I've indicated in yellow. But what we do notice is we indent further and further. We had, for example, a small grain structure, a sliver of grain here, 3. By the time we get to E, significantly into the deformation, we basically have this perfect elastic plastic response. That contrast, that grain seems to, that contrast, I should say, is gone. Does that mean that the grain has been consumed? Is it just not in, the right diffraction contrast? All these things can be kind of sorted out through the upcoming PED data. We can now see that grain number 1 that was in a weakly contrasting condition is now strongly diffracting. And then we can see, for example, grain 2, which is pretty invariant through the entire process, really doesn't change its contrast very much. So we have this great diversity, again, of responses that we have to decipher through our TM images. So what we've done, like we did in the coppers, we do a PED scan before the indent and then after the indent. And so what are some of the really nice features that can come out of this type of correlative work is that, first of all, you can look at the overall texture evolution of all the crystalline grains. And so this is an inverse pole figure that shows the relative intensities, if you will, of different orientations. And so you can see this is the overall, and this is pre-indent and post-indent. What you realize is as I deform the material, we can clearly see that the 001 and 100 textures, as well as 1, become much more pronounced. We can see a shift in some of the preferred textures dominating the 001. And so this gives us, again, this quantitative insight of what is actually occurring in the sample. And we can even break that out even further by looking, for example, at the top crystalline layer and how it deforms as compared to the bottom layer, and then compare that to the overall response. So again, this is just, again, meaning a high overview of what type of quantitative data that PD can give as you're doing these type of experiments. But more interesting is if we can go here and we can look at these various regions of interest and these specific grains. So what we realize is we're going to start here with grain number 1. And if you notice here, grain number 1 seems to have a texture that's close to 113. It's got this pinkish color. But after indenting, the same region, the same grain now has changed its texture to 101. And so this, and it's what it's done is it's experienced a approximately 17-degree clockwise rotation. And then of course you say, well, if this grain changes rotation, maybe that was because the foil itself bent. But because I'm able to do these analysis for individual grains, I can, for example, look here at grain number 3, and I can see that it, for all intents and purposes, stayed very invariant. I can even notice that other grains, so it's grain number 2, which is close to the 111 texture, maintained its orientation before and after the indent. And so why this is exciting now is I can then look at a specific grains, look at its neighbors, and then make a determination, did that grain rotate under the deformation? Was that its response? Before then, I would have to infer that, or if I saw that, I would maybe even have to infer that maybe the whole foil bent, and that's what I'm seeing. But the ability now to go in and individually look at each of the grains and how they themselves are rotating gives real insight of the type of deformation mechanisms that are going on and why specific grains are doing this and other grains are not. And so if I go further now and look at each of these regions of interest, I can again notice that certain grains, like here's 4 and 4, stayed the same orientation, but grain 5 grew slightly. So there's some deformation-induced grain growth, but I also can see that its orientation again changed. And again, and what I'm showing on this side and this side, this would actually be the bright field image. So I'm able to, the PED image, get, I think, improved clarity of where the grains are, where the boundary locates, and then even being able to decipher the type of boundary that separates them. In this case, it's a high angle grain boundary. If I go here to region 2, I can see that this one grain up here, after indentation, has grown and consumed the regions in this area as well. And I can even, again, notice that grain 6, which has this bluish purple color orientation, now has gone to this pinkish red color, indicating, again, a rotation mechanism being along with some form of grain induced growth under mechanical deformation. So even though mechanically induced grain growth has been observed prior to this, most of that work has just been able to quantify, okay, this grain grew, but here we're able to correlate that with its texture and the boundary structures that it forms. And again, if we come here to region 3, we see that in this case, 8, 9 maintain their same orientation, but then they consumed grain 10. And then through our ability to quantify each of the grain sizes, we can pull out the various grain size distributions to look at how much certain grains grew or didn't grow under the process overall in the structure. So what we want to do now is then take that information and actually understand and correlate what we are able to quantitatively do back to models. So the ability to bring modeling and experiments together to help provide a much deeper and richer understanding is really an aim of why you want to do this form of characterization and have these type of techniques. So in this case, Lynn Lee and her student devised out a finite element model structure you can see here that mimics the same sample that we had here. And even though our sample used a cube corner, by looking inside the TM, we can actually see that it seems to be much more rounded than at a sharp point for the indent. And so we then were able to model both the cube corner that we purchased, but we were able to also look at it from a spherical indenter just based upon the shape of what was actually indenting and get a much closer fit to our experimental, much closer fit of the modeling to our experimental response. So now using that information, we're able then to basically realize that we have a slope change that occurs about 17 nanometers in. And at this in-depth, what generates this slope change, because we're able to correlate this now to modeling, is that it appears that the copper layer underneath, the first copper layer underneath the indent yields first, and that results in this slope change. And this occurs at about 0.8 GPa. Then we get to an indentation of 25 nanometers. And at this point, we're starting to see a crater into the sample. And we're able to see that it's initiating now shear transformation zones, because we're not able to actually see this because of all the contrast images in the bright field, but we're able to then from the modeling understand that this behavior now is initiated. And so this is at basically 25 nanometers in, and then using the same model, we're able to go further, and at 32 nanometers in, we clearly see this discontinuity, if you will, or shift, which is related to a pop-in event of a shear band propagating, and it's typically considered a kind of a catastrophic event, and this occurs again at 1.85 nanometers. So this is really nice in the modeling, being able to help us understand what's going on the low displacement curve. But what was really exciting is how the modeling helps us understand the quantitative microstructural evolution that was occurring during the indentation process. As I noted before, is that we saw grain growth that was induced by the deformation, no problem. But we're able to now even look at how the texture of certain grains are going, and the ability to have confidence that grain rotation was occurring as well. Now, mind you, grain rotation is a deformation mechanism that's probably facilitated by the fact that we have a thin foil and a free surface, but nonetheless, we're actually seeing that mechanism and seeing what's going on. And what you can see here, now if I go back to my region of interest 1, we can see, for example, grain 4 is fixed, but grain 5 changed. This gives us confidence again that this is the grain that rotated. It isn't some macroscopic event. It's localized to specific boundaries types. We're able even to quantify the extent of that rotation and its direction, whether it be clockwise or counterclockwise, as I noted earlier for grain 1. We're able to see the same effect here on our region of interest 2. We see that grain 6 is identified here. It's this near a 111 texture, and now we have moved it more towards this pink reddish color. I guess it's moving in a direction towards the 001. And now through the modeling, we're able to look at the stress vectors that occur by the indentation of our spherical indent that we used. And what we realize is to the right of the indent, the stress fields are going clockwise, and then to the left of our indenter, they're going counterclockwise. And by understanding these stress vectors in this direction, we're able to then understand why specific grains were rotating clockwise or counterclockwise in their response relative to their position to the indenter. So again, I like this result because it really ties together the computational aspects of what's going on with the type of quantification you want to achieve in a characterization experiment to fully explain the type of behavior that was occurring. All right, so that's a kind of a correlation of the in situ deformation work. Now I want to talk about how PED can be used in other forms of cross-correlative work. In this case, looking at chemical solutes in specific grain boundaries. So there's a lot of interest right now. Probably it's been going on for at least 20, 25 years, but in the last decade, there's been a significant growth of interest in understanding how to stabilize nanocrystalline grains through the use of solutes placed in the grain boundaries. And these solutes can either reduce the free energy for grain growth, a thermodynamic effect, or they can provide kinetic pinning effects, et cetera. But the reality is that we want to keep the small grains to keep the high strength that is associated with them because of the Hall-Petch effect. The problem is a lot of the models use very basic assumptions in that the solute segregates homogeneously to the grain boundaries and that all grain boundaries are isotropic, meaning they're the same. But in reality, we know that grain boundaries have a diversity in their energy and their structure. We also know from modeling and experiments that solutes in different boundaries can be more enriched in one boundary and not enriched in the others. And this poses a significant challenge because at these small scales, how do we then correlate quantify the chemical structure of the solute, the composition, excuse me, the concentration, if you will, to the specific boundary so that we can better inform our model development. To that end, we want to talk about cross-correlative work of PED with atom probe tomography. So I've already discussed with you to the left how PED works. Atom probe applies a small tip, typically on the order of 50 to 100 nanometers, which is subjected to a high voltage pulse or laser pulse under standing voltage that causes the atoms to field evaporate off the tip. These atoms then become ions, and by their mass to charge ratio, we're able to identify the atom type. And by its impact onto a detector, we're able to determine its XY location by back trajectorying where it came from. And by pulsing the sequence of events, we can then deduce the Z or the Z component. And so what we have then is a set of data that's the mass to charge or atom type, and then X, Y, and Z location. And then we can reconstruct that to identify where all the atoms are. The problem is that these reconstruction techniques in atom probe have some assumptions, which then reduce the spatial resolution. So we're not able to actually, in all cases, clearly identify the grain boundary misorientation. So we can't determine the grain boundary type. But if I could correlate the PED work, which gives me the grain boundary misorientation, with the atom probe work, I would then be able to link these two techniques to give us the high chemical sensitivity needed and the proper high spatial resolution for the boundary identification. And so what I want to share with you today is some of the work we did in iron chromium. And iron chromium's phase diagram is shown here. It exhibits a miscibility gap, so it spinelli decomposes. And we looked in a range between 0 up to about 8% chromium. And what we discovered is when we first deposit iron and we had 4% chromium, we could see that the grains grew. But then once we went to 8% chromium, the grains then became refined. And we wanted to understand why was this occurring. And so when we did atom probe work of a 3% chromium film and an 8% chromium film, we did notice that chromium had a preference to segregate to the grain boundaries, but the extent of that segregation or interfacial excess was significantly larger in its spread at the higher concentrations than the lower concentrations. And the way we're able to figure out this interfacial excess is simply doing a compositional profile, if you will, across a boundary and counting the total number of chromium atoms versus the total number of atoms. And by looking at this discontinuity, it's called the ladder diagram, we're able to then pull out a quantitative value for the interfacial excess. Okay, so with that in mind, what we realized when we go back to modeling, that at 3 or 4%, the actual chromium is actually in solution with the iron. But once we exceed about 5% chromium, then the chromium wants to phase separate out of the system. And so what we believe is occurring that causes the grain size to refine is that the chromium is mostly in solution at these low concentrations, so the grains are able to grow. But because there's a tendency to phase separate at the higher concentrations, they come out, they impinge the growing films on new clusters that grow into nucleuses, that grow into grains, and that refines the grain size. So how does this connect back to PED then? Well, if we look here, we have this large spread. And what we wanted to understand is what is contributing to this interfacial excess spread. To do that, we want to do that cross-correlative technique where we take our sample, as you can see here, we fibbed this work out, fibbed the sample out. This work was done by Rhett Joe. I want to make sure I give him credit for this. Lift out, we thinned it, then we put it into the fib, make an atom probe tip, use a special holder for the TM by Hummingbird in this case, do the PD scan, and then run it in the LEAP to get the chemical information. And then we overlay all this information on top of each other. So here's our bright field image. Again, the PD provides real clarity of where all the grains are, actually even determining what the grain boundary types are that are a little bit more difficult to distinguish in the bright field image. From the atom probe work, we're able to now look at the amount of chromium segregation that's in these boundaries and as a function of the boundary types. And when we do this, we now realize that we see a spread, but we also see that the chromium has a very different extent of segregation tendency depending on the boundary type itself. So for example, high grain boundaries have the highest amount of interfacial excess, where a twin boundary has very little chromium. A sigma 5 has very little chromium. A sigma 3, much higher. And so this now begs the question is what's contributing to why do these differences exist, and what's contributing to even the experimental spread that we're seeing in our analysis. And again, our application of cross-correlative work to PD has been really helpful. Up to this point, I've only talked about grain boundaries in terms of their misorientation, right? And that's just three degrees that we've been able to identify. But to properly quantify a grain boundary, you need to know all 5 parameters, i.e. you really need to understand and know what the grain boundary inclination plane is. Now in PED, and there's a really nice paper by Kiss and Labar for microscopy and microanalysis in 2016 that talked about using PED, and as you go across the grain, you'll start to get an overlapping of the diffraction signals or diffraction patterns from grain 1 and grain 2. And you can weight average those to get an idea of the approximate inclination of the boundary. The problem is that boundary could go from, in this case, left to right, or alternatively, the signal could be going from right to left because it's a 2D projection. But Atom Probe is a 3D reconstruction, and so by looking at this signal, looking at the 3D reconstruction, we're actually able to decipher the actual inclination angle in its proper form. And in doing so, we're able then to look at how the interfacial excess, the chromium solute, varied as a function of a particular grain boundary type and its inclination angle. Again, we couldn't pull that type of information out of just an Atom Probe run, nor could we pull that information out just from a PD run, but the combination of those two really gave insight of why there's a spread in the chromium excess in the different boundary types, even though I say it's a sigma 3, but we see a certain spread. And what we learned is that as the inclination angle increased for a sigma 3 boundary, the chromium content would go up. In other cases, like a sigma 5, it was relatively invariant, and in the sigma 9, it had an inverse relationship, as did the sigma 11. Using that information and using a hybrid molecular dynamic and Monte Carlo simulation, we computed the same, what would we expect for those particular inclination angles and our chromium contents. And what was really nice to see is though the excess values themselves there's some discrepancy here, these are much lower than here, the relative trends were reasonably in agreement that a sigma 3 shows an increase in chromium content with inclination angle that we were able to decipher through the correlative aspect of Atom Probe and PD. The sigma 9 showed this inverse relationship, which we saw experimentally. So, and then we were able to take the computed or calculated values and then compare that to our measurements that we did experimentally, and we saw reasonably good agreement. In this case, sigma 5 was low because it's an anti-segregating boundary. Sigma 3, we can also see that, a very good agreement. So we felt really, I thought this is a very nice example again of how correlating your, in this case, Atom Probe experiments with PD can help you give improved understanding of why materials behave the way they do. Now, of course, we're left here. Why does the high angle grain boundary show the largest spread? And what we realize in these cases, both from our simulation experiments, the chromium in these type of boundaries tended to not wet the boundary as much as cluster. And depending upon where you made your measurement, you could see a high or low concentration. And so it was a lot of it could be user dependent. And it was just innate to how chromium behaved into that sample. The last example that I want to share with you today is how we can actually do in situ heating experiments with PED. So in this case, we use this dense chip. Now, prototype has one, but as well. But the idea of these devices is that they can provide rapid heating, but more importantly, they only heat up a small thermal mass, meaning that the samples don't drift. So in principle, you can do a PED scan at temperature. Now, in our group, we tend to heat up and then cool down because it goes so quickly, and we can stay in the same region of interest and do the scan. But we have done some experiments where we can heat up and do the scan at temperature because of thermal stability that occurs. So in this case, we look here again at our iron chromium sample at 4% and 17%, and this is kind of the heating sequences that we did. We started at 400C and 800C. Now, the original intent of the experiment was to understand grain growth, but like many experiments in life, that after you get it started, you kind of come across other features that you didn't anticipate. So just in summary, by doing PD, I'm able to again determine texture, put out grain sizes very easily, no problem. But what was really fascinating was once we got to 700C, we started to see the onset of the formation of chromium oxide in the higher chromium content film. Now, we didn't see this in the 4% chromium film. It's because the chromium stays in solution. But in this case, where the chromium then is being pushed to the grain boundaries, it has a higher affinity than iron for oxygen. And even though I'm in a TM that's under vacuum, it still has a partial pressure, though low, of oxygen. And that actually starts to react with your chromium. But being such a low partial pressure of oxygen, it also allows the oxidation to occur in a much more slow and controlled process. And so despite there's been numerous and extensive studies on oxidation, capturing the early stages of oxidation and its effect in evolution in microstructure can be quite daunting and difficult. And again, what we're able to do here is to do this. But more importantly, through the PED, we can actually understand and quantify the boundary types, where they're forming and how they're evolving. And so this is kind of the last example I want to share with you today. What we're going to do is look at this particular region that you can see here highlighted by this yellow box. At 700 degrees C, we can see this junction and there's no chromium oxide. Once we went to 800 degrees C, we see that the chromium oxide particle phase precipitates out of the boundary. We can even see along one particular boundary additional oxides forming, but on the other boundaries not. And again, from a bright field image or even dark field image, it may be deducing, well, why is the chromium, for example, forming on this boundary and not on these other boundaries? How did this type of boundary, where there's almost like 4 of them coming together, but it nips together into one, how did that evolve into this structure? And so what we can do now is look here at our grain boundary type map. And what we see then is that our initial boundary involves 3 high angle grain boundaries that are, 2 high angle grain boundaries. 1, 2 comes up into here, and then 4, and 4 is actually a sigma 5, a special character boundary. And then after it oxidizes, we can see that it removes grain boundaries 2 and 3, the high angle grain boundaries, but still keeps grain boundary 1. And in the process of these boundaries being consumed out by the precipitation, this sigma 5, this number 5 boundary, which is a low angle boundary, then comes up from the back end. So by knowing the boundary types, we actually can then deduce what's going on. These high angle grain boundaries likely provide the fast track diffusion paths for the chromium to meet at a triple junction and then to react. And the fact that this is a high angle grain boundary here, and we see chromium, probably has a higher chromium content, which we've already seen from the cross-correlative atom probe work I've shown, as compared to a special character boundary, meaning that the higher chromium content allowed this boundary, a high angle boundary, to precipitate out additional chromine precipitates, where these boundaries didn't show this, and it's because they were a special character boundary that was revealed through the use of the PED work that we did. Moreover, now that we know the boundary types, we can actually determine the dihedral angle between the boundaries. And we can see here that in this case, the high angle boundaries compared to the special character boundaries is a lower boundary, indicating that it's probably the more preferred boundary. It's probably the more rapid growing boundary for the oxide as compared to the other two. And then finally, by having all this information, knowing the boundary types, we can even use Young's equation to get the relative interfacial energies that exist between the different types of boundary types and the oxide structure that forms. And this is just shown here. It doesn't give you, obviously, the interfacial value, but the relative value of how they can compare. And again, what you see is that you can get a deeper understanding of how specific boundaries may be contributing to different forms of oxidation and how that evolves in a microstructure. I know that I've covered a lot of different examples. Again, I hope I gave a kind of a high view perspective of how PED or ACOM, this automated crystallograph orientation mapping, can be used through in situ microscopy to help reveal structures that may be masked through diffraction contrast or correlated with other methods that bring the sum of the two individuals is larger than the contributions of the individual components themselves. And I hope these examples illustrate those issues. One of the advantages of PED is that it has the ability to probe really finer length scales that are only a few nanometers and takes more previously qualitative electron-based images to quantitative that can be to modeling and simulations. And then finally, I think the whole coupling, this whole in situ TM offers a really exciting and new area of where all these techniques can kind of come together and support each other. Again, I want to thank you for your time.